Okay, so we talked about various components of the formulation, drug formulation, and how they will stabilize your drug. Okay, so the overall goal is to increase the stability of your API molecule before your drug is injected to patient. That's why you have to formulate, and that's why you have to add those different components in your formulations to extend the stability of your drug. Okay, so it's the same topic, but let's talk about this in terms of how do we increase or optimize the stability of your drug. Okay, so drugs in solution are subject to major mechanisms of degradation, such as hydrolysis. So hydrolysis is what your drug is going through a chemical process. So water molecule is broken as well as your API is broken. Okay, and what other damage is possible? Oxidation, as we already said, and then exposure to strong light that will develop photolysis. Light energy-based drug molecule lysis is the photolysis, and then covalent and non-covalent aggregation. So by forming covalent and non-covalent bonds, your API will aggregate, kind of randomly forming big chunk garbage. Deamination, amino group is taken off from your API. Cleavage, just cutting up your API molecule at random site. And surface denaturation reaction, like I said, drug surface can be denatured if it's not stabilized. So those are the main cause of instability of your drug API molecule. So then, what do? You need to optimize hydrolytic stability so the hydrolysis doesn't happen. Okay, so how do you prevent hydrolysis happening from your drug molecule? Okay, so by optimizing hydrolytic stability of your drug, you do that. How do you do that? How do you optimize it? By maybe you have to look at the sequence of your drug API molecule. So if your API is a protein molecule, of course, proteins are made out of amino acid, right? So it will have a sequence of amino acid, and you will see if your protein sequence is prone to hydrolysis. If there are certain amino acids which are prone to that, then you may want to remove those amino acid sequence and change it to something else without losing the structure and function of your drug. Okay, so that may be done genetically, genetic engineering. Okay, amino acid sequence change, yeah, that's done at the DNA level. And then you can formulate at optimal solution pH, of course. We already talked about why you have to do that. And you may want to store your drug at low temperature, of course. So the reactions will slow down at the low temperature. And optimize the effect of ionic strength using empirical approach. Empirical approach is what? Scientific approach, by testing and see and experiment through various experiments, you find the best approach. That's what empirical approach is. Okay. And then, of course, buffers and hydrolytic stability. So buffer is needed to prevent hydrolysis. Of course, maintaining the same pH is important. We already talked about that. So buffers are made out of salts and some ionic compounds. Good well-known buffers are like acetate, citrate, phosphate, okay, or amino acid can be used as buffer as well, okay, in maybe monoclonal antibody formulation. The ionic strength is another important thing because they protect the drug by forming good electric field within the solution. Okay. So increase the ionic strength would be expected to decrease the rate of degradation. Okay, but you don't want to add too much. Yeah, so isotonic, like I said, is good. Okay. And then what else? Optimizing oxidative stability. So to prevent oxidation from happening to your API, okay, you need to optimize the kind of a stable condition within your formulation. That's what it is. So how do you do that? By adding like phenol or catechol or thioethers, and they can be susceptible instead of your drug if you add those guys, so they can protect your drug from oxidation. Okay, so epinephrine, dopamine, morphine, tetramycin, small molecule drugs that will readily oxidize in solution. Okay? And protein drugs that has amino acid sequence of methionine, cysteine, histidine, tryptophan, and tyrosine. These amino acids are well oxidized. Okay? So they are susceptible to oxidative damage and photolytic damage. At the strong exposure to strong light, they will be also oxidized. That's what it means. Oxidation of sulfur hydrate containing amino acids like methionine and cysteine will also lead to disulfide bond formation and lose the structure of your drug and biological activity of your drug. So those are, amino acid sequences that are prone to oxidation damage. You need to know that. Example is human growth hormone, chymotrypsin, lysozyme, parathyroid hormone, human granulocyte, colony stimulating factor, insulin-like growth factor, monoclonal antibody, OKT3, interleukin beta 1 beta, and the glucagon are exemplary drug that can go oxidative degradation. So you have to be concerned with their special formulation. Okay, so how do you prevent oxidation of your drug? By adding these guys like antioxidants. Okay, ascorbic acid, sulfurous acid, thioglycerol, and the thioglycolic acids are frequently used. And then we probably talked about before DTT in different classes, dithiotrenitol and glutathione. Those are naturally made reducing agent found from cells, and mercaptoethanol. There are two readily oxidized, so they are not practical in pharmaceutical formulations, although they may be good for your experimental formulation like in vitro, but not good in pharmaceutical formulation. That's what it means. Okay, especially if it's requiring long-term storage, they are not very effective. So do not use DTT, glutathione, mercaptoethanol, those kinds. Okay. And then chelating agent, we already talked about it. Okay, metal binding substance. Okay, to inhibit free radical formation, especially it can suck out all those transitional metal ions. Okay, so copper ion, calcium, magnesium, manganese, and zinc. Okay, those ions could be removed through chelating agents. What are the examples? Like EDTA, disodium EDTA. Okay, the final concentration should be lower than 0.4%. Okay, and the thing is, it doesn't go into solution very well. So first ingredient should be dissolved during the compounding, because it doesn't get dissolved first. So this should be dissolved first, and other things should be added. That's what it means. If you have the protein that it contains metal ions like insulin, hemoglobin, fibrillase. Okay, if your API has metal ion in them, you don't want to use metal chelator, because metal chelator will suck out the metal ion which are required by your API molecule to maintain the structure and function. So that's why the chelating agents would not be good for them, right? So you better not use a metal chelating agent if your API has metal ion in them. Makes sense. Enoch gases, oxygen is, the source of oxidation. So instead of oxygen, you can fill your drug vial with enoch gas. That's what we talked about before. Packaging and oxidation. So brown bottles are good if your drug is susceptible for photolysis, blocking the strong light. It could be, effective. And then if oxygen is transmitting through your primary container, that's probably not good. So sealing should be well done. Okay. So, and then oxygen transmission coefficients are determined by the manufacturer of the stopper. So stoppers should be well studied. So there's no gas exchange. That's what it means. And then in brown bottles, the amber colored primary packaging material is used if your drug is susceptible for light damage. Makes sense. And then other stabilizers minimizing drug degradation: sugar, glucose, dextran, or polyols, ethylene glycol, or glycerols. Those can inhibit metal catalyzing oxidation. Okay. So they can chelate metal ions or scavenge free radicals. So you can add those. Among those, mannitol can inhibit the ion-catalyzed oxidation of methionine-containing peptides, and that's most commonly used excipient in freeze-dried formulation. So you will do a lab for your formulation process. One of the formulation you will use for that lab would contain mannitol. So we'll talk about that again during the lab. And then co-solvent like glycerol could stabilize protein in solution, as they are excluded from the surface interaction within the protein. So you could add glycerol in your formulation. That's what it means. Optimizing physical stability. Okay, so many proteins are unfolded when exposed to stressful physical condition, like temperature fluctuation. Okay, exposure will make hydrophobic portion of a protein become exposed to aqueous environment. Then you'll form aggregation. Okay, it's like boiling same way. Then physical instability and a loss of biological activity. Your drug will lose its structure and function. That's not good. So denaturation is a process. Exposure to higher heat, then your protein will be unfolded or misfolded. So that's not good. Once your protein is denatured, normally it's not reversible, but sometimes it can be reversible. But if it's reversible, that's good. But if it's not reversible, it's becoming aggregate. That's a problem. So you don't want that. So aggregation you should not have with your drug, because aggregation meaning your protein is, binding together, is forming a big garbage. If that big garbage is injected into human body, it's going to cause anaphylaxia. What is that? Severe allergic reaction, because your immune system will recognize that as a foreign substance and try to attack that, and it will cause hyperimmunogenic response in that area. So you don't want that. Non-specific hydrophobic interaction is the reason why they have that. So that may be caused by exposure to air liquid and solid liquid interface, and then light and temperature fluctuation. You probably don't want that. So keep your drug, of course, in a constant environment. That's the best thing. And impurity you should not have in your drug. Foreign particles you should not have in your drug packaging material, packaged drug or filled drug container. And irreversible aggregation can be minimized and then prevented with formulation involving stabilizers. We already talked about it. Okay, so this is showing you, if it's a folded your drug structure, reverse the unfolding. Okay, may come back. If it's going back to the normal condition, normal structure, it's a reversible. Okay, for misfolding and unfolding or folding, that's not bad if it can be folded back. But if it cannot be folded back, that's irreversible, which is good. If this is not good, so it is unfolded drug. If they come together, you'll form big aggregates, which is not good, which will cause allergic reaction in the patient's body. Okay. So pH should be maintained, as we already know. Example, 0.125% of alkyl saccharide excipient that can stabilize and reduce aggregation. So that's important at some point. And absorption we already talked about. Drug is binding on the primary container, so it's not good. So you can add more protein, so if it's absorbing, so maybe part of it. And filtration, siliconation, surface, yes, I already talked about it. Okay.